Hey, yo, is this thing on? What up? It's Rutherford. We are here doing Unit 4 multiple choice questions for my APbio class. I'm going to set this up and we're going to rip through the evens. Okay, so we got number 2 up here on the screen. Cell cycle. Okay, so Unit 4 is mostly mitosis. There's some other stuff too, but all stuff that has to do with that. So pictures, graphs, knowing how to read a graph is really important. So I'm going to skip down to the question because a picture says 1,000 words. So you can look at that graph for forever and not really know exactly what you're looking for. Obviously, we can see some stuff happening in prophase, metaphase, anaphase, telophase. Okay, so mutant 1 cells are more similar to mutant 3 than to wild type. So now I'm looking up, I see the key here is going to be wild type and mutant 1, mutant 2. So mutant 1 is the dark one, is more similar to mutant 3. Okay, so these two, they don't really seem that similar at all. I'm just going to move away from A. I mean, I don't know anything of what they're talking about. I'm just like, those bars aren't even remotely close. Okay, B says wild type, which is our first bar. The percent of cells in anaphase is twice the amount of those in telophase. Okay, so are we talking about percent of cells? Sure, yeah. So now I'm finally looking at the y-axis, percent of mitotic cells, the number of cells that are actually going through mitosis and dividing, making more, rather than the other cells that are just like specialized and working, differentiated G0 cells. So wild type percent is twice as much as in telophase. So I'm looking at wild type here is around 52, and then in telophase there, anaphase to telophase. Oh, no, there's no way. Total trash. Okay, so B is out in C for mutant 3 cells. Over here, more time is spent in prophase than in other stages. No, I mean, that is true, but that has nothing to do with this graph. So I do like it better than A and B, but I'm going to move on to D. The percent of mutant 2 cells in anaphase is higher than that of mutant 1. Okay, so this is basic. Let's look at anaphase. Let's say mutant 2 higher than mutant 1. Mutant 2 is right here. Mutant 1 is dark. And yeah, so that's not true either. So I'm really going to go back to C. If I got to go quick, honestly, I ruled out A, B, and D pretty strong. Maybe I could flag this question because it doesn't really make sense there. But I'm going to feel comfortable with C. And I'm going to rush and just ask for the answer here. Boom, Shazam. Okay, so interestingly enough, that required you to read the graph, but the answer C is not about reading the graph. You have to kind of rule out A, B, and D by reading the graph, but answer C is just something we get from our knowledge of mitosis. All right, number 4. Too easy. G1, G2. Okay, so the cell cycle goes G1S, G2M. M stands for mitosis or meiosis. The expected percent change in DNA content from star G1 to G2. This is just basic knowledge, knowing that S phase is DNA replication. And so when you're going to replicate your DNA, you're going to have twice as much DNA. Now, I know this might sound weird, but in math, if you have twice as much, that is 100%, right? So if you've got 100% and then you replicate, now you have 200%. I know that sounds weird, but that's why we know D is the correct answer here. Okay. Nice and easy. That was a softball. Come on, give me something harder. There we go. Okay, again, there's so much stuff I could read up there at the top, but like really, we got to move faster. Okay. Oh, this is a multiple choice question. Or I'm sorry, this is a free response question. Okay, so that's not my focus of these videos. So we're just going to keep moving. But again, notice it's just reading data, graphs. I will spend some time on those in the future, but not this time. Now, this little line up here at the top, 7, 8, 9, 10, 11, indicates that all of those are based on the same thing. So you'll notice that the beginning for all of those, it's the same picture, same thing with this G protein coupled receptor, which we haven't talked about yet, but we will. This is just a basic membrane protein. So you can kind of see there's a plasma membrane, and then there are these gray things, the beta-2 adrenergic receptor and the adenylyl cyclase. Don't be worried about those kinds of things, okay? You can look at that and say receptor. I know what a receptor is. And adrenergic, it sounds like adrenaline, okay? Epinephrine, like an EpiPen. might sound like adrenaline. And then cyclase ends in ace, so it's an enzyme. So we're not like too far off on this. We don't really need to know exactly what's going on. We will just kind of see that, oh, look, like whenever we have this big diagram, we draw these diagrams and a variety of things for biological systems. We have an input and an output. There's all these steps in between because evolution takes many steps. It doesn't take the shortest route. So basically, we're taking epinephrine and we're getting glycolysis out of that. Okay. Our memory of glycolysis is glycolysis. So we're cutting glucose, okay, to get energy from it. This is right before the stage in cell respiration. And it says, even if you want to read this, epinephrine, muscle cells, energy, ATP. We got a lot of mitochondria there. So now, I mean, I can go over that stuff for you guys, for the benefit of you guys, but really, if I was doing this, I'm just skipping to the question. Which of the following statements best describes the role of adenylyl cyclase in the epinephrine signaling pathway? So this signal pathway drawn with this leads to this, 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 signaling pathway. Adenylyl cyclase right here, it is, you see right there, ATP goes to cyclic AMP. So we know what ATP is. Maybe you don't know what cyclic AMP is. Okay, I could teach you that. AMP is a adenosine monophosphate. So it's only got one phosphate. ATP is adenosine triphosphate. It's got three. And you're like, where the hell is the second one? ADP. That's what we usually talk about in class. Forget it. Okay, we know what ATP is. Even if you don't know what AMP is, you know that's what adenylyl cyclase does. Okay, so let's see if we can find the answer out of that. It converts a polymer to its monomer subunits. Now, listen, ATP does have three, and AMP, maybe you don't know, but we know ATP is not a polymer, okay? There are three phosphates, but it's not like you have an AMP, and another AMP. You have the A, and then you can add three phosphates to it. So it's not really a monomer polymer situation there, okay? That's really only for carbs and proteins and nucleic acids. We've never talked about ATP being a polymer. So that might just be able to rule out that. B says it moves substances across the plasma membrane. Literally, if you're just looking at this thing and seeing, like, a 10-year-old should be able to say, no, ATP going to cyclic AMP, that's not going across the plasma membrane. It's all on the inside. Okay, it's just on one side. So I don't like that. C says accelerates the production of the second messenger. Ooh, gosh. I mean, it does seem like there's a bunch of stuff happening at the membrane here between the beta-2 adrenergic receptor and the adenylyl cyclase. It does seem like there's a whole bunch of stuff there with GTP and GDP. And then all this stuff with the protein kinase and the phosphorylase kinase and all the laces there. That might be good because it does seem like a second messenger, as in the first messenger is epinephrine and the second messenger is cyclic AMP. That is a little bit higher level, and we would want to practice that and memorize that. But I'm pretty sure that's the right answer, but let's look at D. Phosphate groups from ATP to protein substrates. Ugh, gosh, that's a pretty good one because we do have phosphate groups, and the protein substrate would be adenylyl cyclase. Oof, that's tough. That's a toss-up between C and D. I would flag this one. Not entirely sure which one it is, but I think C is correct, and D is just like, that's a very solid answer. Like, we have ATP going to AMP. Therefore, where did those two phosphates go? They probably went to adenylyl cyclase, which we know is a protein. I'm just going to have to guess D. And I might get that one. Yep, I got it wrong. Finally, great. Okay, so the production of a second messenger. Okay, so at least I narrowed it down there. And if I had more time at the end, I would go back. All right, so we'll stick with this problem. We're going to go to number 10 to see if they can stump me again. Okay, this is some tough stuff. This is chapter 11 stuff, and we'll have to go over this because I usually say this for the end of the year. In fact, we haven't studied it yet in our mitosis unit because this stuff is so difficult. I save it right for the end, so it's really fresh in your mind. Okay, this cell signaling stuff, which is pretty cool, and you just kind of have to, they use this for 5 different problems because this is kind of just how it works. There's protein kinases, and there's G protein coupled receptors. And once you know those two, That's kind of how cells communicate. You know, you can either knock on the door or you can ring the doorbell. That's kind of the ways that you communicate with someone from outside of the house to the inside. And for cells, it's really just kind of two ways. Now they're complicated in the way that they work, but we can figure them out. Okay, so I'm already comfortable with this, so I'm going to go right to the questions. Epinephrine is a signaling molecule. Hell yeah, that is totally correct. If I have no time, I'm like, hell yeah, that's the right answer. B says G protein in the epinephrine signaling pathway consists of 3 different subunits. Well, yeah, okay. Oh, here's where if I see 2 things that are true, I'm like looking back at the question. A researcher claims that the epinephrine signaling pathway controls a catabolic process. So catabolic, we're thinking release of energy, okay, exergonic, and I would draw that right here, okay, exergonic, okay, it takes half a second, and it extends your mind onto the page and gives you something to look at and think about. Okay, so we want something to justify the researcher's claim, okay, so to me, B has nothing to do with epinephrine or the catabolic process. The fact that G protein is 3 subunits rather than 2 or 4, that really doesn't matter. That has nothing to do with epinephrine actually initiating this whole process. That's kind of like the fact that if you have a glass door or a wood door, you can knock on either one, you can ring the doorbell, like it doesn't really matter what the structure is of it. So I don't like B. Let's try C and D. Phosphorylase kinase catalyzes the hydrolysis of ATP. Hydrolysis, cutting ATP down to ADP, and in this case down to AMP. But the phosphorylase kinase, oh, it is right there. Cool, phosphorylase kinase says ATP. That's true, but again, this question is asking about epinephrine, okay, controlling the catabolic process. That really has nothing to do with this little step in here with phosphorylase kinase. It doesn't, okay? Phosphorylase kinase is just a part of this cascade, a part of this pathway. It's not actually the initiator. It's not the point. It's not the product. So I don't like that either. And then our final one, glycogen phosphorylase, right there, catalyzes the conversion of glycogen to glucose 1-phosphate. Ooh, yeah, so that one, we have an enzyme and we are going glycogen, which if you don't know, you can think, oh, it's probably like glucose, that leading to glycolysis. That does talk about muscle cells, and that is the result, okay? So A and D are my best answers at this point, okay? The researcher, so I'm going to go back to the question just to check, because it's always good to go back. Epinephrine signaling pathway controls a catabolic process in the muscle cells, which best helps, okay? Obviously, A and D both help. Which one best justifies the claim that epinephrine controls a catabolic process? I really think it has to do with D, because that's the whole deal in terms of catabolic muscle cells. Like, what's the point? Epinephrine can bond to a transmembrane protein, but that doesn't necessarily mean anything's going to happen. However, if we know that something is happening in the end, that's what can justify the researchers like, okay, so we see epinephrine and then we see glycolysis, the glucose 1-phosphate. Okay, so I really, although A is the start, D is the finish, and that's why I think D helps support the process. Good, cool. Okay, nailed it. All right, now we're on to number 12. Make a different question. Oh, cool, another signaling pathway, significantly easier. Don't be scared by the tough ones, okay? Just dive in. You got to break it down. You know, notice how like I didn't spend my time really focusing on all the little steps in the signaling pathway. Just go right for the question and the answers and see which part of the pathway you need to look at. You don't need to like understand. Kids want to like, sometimes people want to understand everything completely before they even try the problem. That is like the wrong way to do any AP test. Wrong, 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 wrong. You just got to like freaking dive in and go for it. You just got to try it. Okay. All right. Okay. So I'm going for questions. More, less, less. Again, when you go to the answers, you can kind of see what are they looking for. So if I feel like more of this stuff or less of this stuff will be secreted from the kidneys, so that middle part is the same. So if I can draw on this, I'm just going to, for the mores, I'm going to write up. For the less, I'm going to write down. That middle section, I'm just going to cross out because there's nothing different. All those are the same, secreted from the kidneys. Then we have increasing, decreasing, increasing, decreasing. Okay, so this is a classic, just like, find whether it goes up or down, and then does that cause up or down? Okay, so if you don't know anything about erythrocytes and erythropoietin here, we could just kind of look at this and just see the arrows. The arrows say low oxygen levels in blood stimulates secretion from kidneys. So that's right there, okay? Then that stimulates the erythrocyte production in bone marrow. So I'm looking at that pathway, and because I looked at the questions, I'm like, I'm pretty sure that's what's going on. Now, I haven't even looked at the question yet, okay? I could double check the question because it seems kind of easy to me. Low means secretion, means production of erythrocytes. So I'm like, okay, erythropoietin, more of it will be secreted by the kidneys, and that increases production of erythrocytes if the oxygen is low. So B seems correct to me. If I'm going fast, boom, that's exactly what this thing says. I know I didn't look too much at this. So if I have time, I'm gonna double check it. Air is less dense at very high elevations, less oxygen, right? Okay, so this is altitude sickness and altitude training. If a person travels from sea to high elevation, that means there's low oxygen, okay? And boom, decreased blood oxygen. So it's gonna be B. I'm feeling pretty confident here. And then you got to move on. When you're feeling the confidence and you know what's going on, this is my specialty, like I study this stuff, I know about this stuff. So I just got to go with that. Now let me just kind of explain it for a second so that we're comfortable with these concepts. Okay. Blood is produced in the bone marrow. Okay. And there is less oxygen altitude, and low oxygen means that you need oxygen, and what carries oxygen is red blood cells. Red blood cells are called erythrocytes. Erythro means red, and cyte means cell. So erythropoietin is secreted apparently from the kidneys. I didn't know that, but your kidneys filter your blood, and so I can imagine that if your kidneys are filtering your blood and they're like, "Yo, we don't have enough blood, enough hemoglobin, enough erythrocytes, red blood cells," then it's going to want to make more. Okay. Now, if then that increases the oxygen levels in the blood, then it can tell the kidneys, hey, we're good. Okay. And then it will turn it off. So this is a classic like negative feedback cycle here, where like if your house gets cold, then your heater kicks on, and then it warms up, and then your heater turns off. If your house gets too hot, the AC turns on, and then it turns it off. So it's always trying to maintain the right level of temperature, and in your body, you're always trying to maintain the right level of red blood cells. So if you change your environment and there's low oxygen at altitude, then you just need to make more red blood cells. Pretty neat. Okay, let's on to number 14. Okay. Yes. Okay. Again, another signaling pathway. That's just like the name of the game here in Unit 4. It's mitosis and signaling pathways. It's pretty simple here. The general concept that I can just say before we get into the question is like, you got stuff knocking on the cell, and then you got cells making proteins or like doing something. You got to have enzymes doing some sort of reaction and responding to whatever is knocking on the cell membrane. Okay. And that's usually something that's going to, here, it looks like this glucocorticoid goes straight in. It doesn't bond to a membrane protein, which is kind of interesting. And then a bunch of things kind of go together, and we got protein, and then it gets into the nucleus here, and we have gene expression, which is transcription, translation, RNA polymerase, ribosome making a protein. Okay. So let's look at the question. Which of the following best predicts the effect of a mutation that results, again, this is a malfunction thing. Okay. So malfunction mutation is kind of like, what happens if this breaks? Okay. A breaking question. In the glucocorticoid receptor's ligand binding function. Okay. So even if you're not comfortable with the word ligand, you do know what a receptor is to receive something. And so let's find the glucocorticoid receptor. There it is. Okay, so there's an activated one and an inactivated one. The ligand, I will tell you right now, is just whatever binds to the receptor. Okay, so if you're catching the football, you are the receptor and the football is the ligand. Okay, this ligand is the glucocorticoid that's coming in right here, this little triangle, and it's going to land right there in that perfect spot. And then it's active, and then kaboom, then it can enter the nucleus. Without that, it can't enter the nucleus, and it wouldn't be a transcription factor. This could bind to like the operator region, or could take off the inhibitor or something, kind of like the lac operon or the trip operon. We could cause some sort of gene expression. So that's what we want to kind of be picturing here is the operator regions, the promoter regions, and cool. So this glucocorticoid. So to the answers, if something is broken. So I would like literally draw on this thing if you can. You're like a loss of the receptor ligand's binding function. This is pretty simple. If you can't bind, you ain't going to make no genes. Well, not make genes. You're not going to express the genes. Expressing the genes means making proteins. So if it doesn't matter if glucocorticoid is there, you're just not going to make a protein. So that's what I'm looking for. The rate of diffusion, concentration of glucose, and the glucocorticoid receptor, transduction of the cross, it's D. I know it's D because D is the only one that says nucleus. Nothing says that the glucocorticoid can't come in. It's just not going to bond. Now, these accessory proteins are going to stay, it's going to stay right there. So you could maybe circle this part and be like, stay. And then you could cross this out and you could cross this out and say, no, or just cross those out. So, that's not going to happen. So as soon as D says the concentration of the receptors inside the nucleus, well, yeah, nothing's going inside the nucleus. We don't have the key. We don't have the football in the receiver's hands, and therefore we can't go to the end zone. I know it's D. Oh, they got me. Cool. Got it wrong. So the receptor will remain associated. That's true too. That's true too. Oh, I didn't see the word increase. That's why it's also very important. See, as I'm like talking through this and going fast, I'm not as focused. I'm missing those keywords. Okay. When you see keywords like mutation and loss, circle them, underline them, draw an arrow for loss or an X for loss. When you see words like increase, up, you could just scan over the whole thing. And if they're doing an increase, decrease kind of a question, which is like pretty common, whenever something's broken or mutated, you got to look for increases and decreases and stuff. And that's where they got me on D. No worries. Remember, you don't have to get 100% on the AP test to get a 5 or a 4 or a 3. You don't. You just have to do better than average people. And you have the tips and tricks because I'm teaching you these little arrows and circling and crossing things out and writing stuff down and being able to go through these questions intelligently and it's not about like who knows the most APbio. It's about who knows how to take this test. Okay. So as long as you take this test better than most people, you're going to be passing. That's how it works. This is graded on a curve. So don't worry about getting every question right. And don't fret. Like I'm not worried that I got those questions wrong, right? I don't want you to be worried about that either. If you feel like you get to a question, you're like, I have no clue, and you just toss it up, just move on. Very, very important to just move on very quickly. Forget those other questions. So boom, moving on. Vertebrate immune systems, communication, short and long distances. We know that, okay, electrical and chemical. And how cell surface proteins such as MHC and T receptor mediate cell, we haven't talked about immune system or chapter 11, but let's see if we can still answer this without that knowledge, okay? We know what a protein is and we know what a receptor is, period, okay? And cell communication over short distances. We have talked about how short distance stuff, communication. So let's try. The proteins receive electrical signals from nerve cells. That's long distance kind of stuff. Okay. The proteins leave the cell and travel in the bloodstream. That's long distance. And the proteins interact directly with proteins on the surface of other cells. That sounds pretty nice. From cell to cell, protein to protein. Okay. One release here. It's not some big across your body with bloodstream or, a huge nerve cell coming from your body or back to your brain and spinal cord and then back out. So I like C. If I'm rushing on this, I'm going C. Let's try D. The protein binds to molecules secreted by cells located in other parts of the body. Okay. other parts of the body. Again, that's long distance. I mean, I can't, and again, here's where I'm like, ooh, but maybe I should try because I got 2 questions wrong so far this time. So maybe I should really sit down only if I have time. Okay. Do not let your problems with previous questions, maybe it can remind you to like slow down a little bit, but don't let it crush your confidence at all. I am feeling like this is an easy ass question and I have got to go with C. Shazoo. Okay. Boom. You cannot let your confidence be fooled. You will get questions that are like, my God, I have no clue. Okay. And it's not going to tell you which ones you get right and wrong. Okay. So that's kind of nice to maintain your confidence there. But when you feel like you got no clue on a question, keep moving. Okay. Keep that confidence, keep moving. You do not need to get 100%. I don't even know what the percent is, but it's like, I think if you get in the past, it's like you can get a 60, 70% on the test, and that could be a 4 out of 5. Like passing is a 3. And that means you might get like 50%. So if you're going to like half the questions right, you could be on pass to or on track to passing. Okay. So we're shooting for more like 75%. So we just got to like, focus on keep moving and keep our brain fresh. So the cancer cells behave differently. Oh, duh. All right. So which of the following conditions most likely cause to become a cancer cell? All right. The environment contains cancer cells. Oh gosh, we just got no cancer details. I don't know if there's any way to get around this. Let's just pretend you don't know anything about it. The environment has an abundance of nutrients. The environment lacks signals that would otherwise tell the cells to stop dividing. Sounds pretty nice. Okay, the environment contains mutagens that induce mutations that affect cell cycle regulation. That also sounds pretty nice. Woof. Okay, I'm going to go back to the question here. If I'm going fast, I like C. It just seems pretty nice as far as cancer. They ignore signals. Which of the following conditions would most likely cause a normal body cell to become a cancer cell? I do like C, A is like, cancer cells don't really convince other cells to be cancerous. Cancer cells just kind of like go through mitosis and make more of themselves. They're not really converting other cells. They're making more cells. Get it? B, abundance of nutrients. That's not what causes cancer. I mean, not really. C, the environment lacks signals that would otherwise tell the cell to stop dividing. I really like that one. That's pretty solid because the other cells usually tell the cancer cells like, yo, bro, you should stop. And then the cancer cells don't listen. But D is kind of nice too. The environment contains mutagens that induce mutations that affect cell cycle regulating proteins. That's pretty good, but I like C a little more because right in here, it's telling you ignore signals that tell them to stop dividing. Okay, now this is a cool thing about the AP test that you should know. If the answer is C here, they told you, they're telling you that they ignore signals that tell them to stop dividing. So you look at C and it goes, the environment lacks signals that would otherwise tell the cell to stop dividing. So it's kind of like they just rephrased it here. Oh, okay. And it's wrong. So yeah, there's a half and half there. Great. Okay. I thought that was a really good one too. So that one totally works. I can't necessarily explain really quickly why the difference between C and D, but let's move on. We're going to go over to 20. All right. Got a few more. Flowering plants, plasmodesmata. Oh God, what are those? Adjacent cells, if you know that word from SOHCAHTOA in geometry. An explanation of how plant cells communicate across cell walls. Okay, again, our cell communication thing is we've got molecules landing on membrane proteins. If we just remember that concept, we'll get really far. Okay, so the question is, the diffusion through plasmodesmata of which of the following? Okay, we're talking about cell communication. Do cells communicate by sending each other organelles? Like, hey, cell, I'm going to send you my entire mitochondria. I'm going to send an entire lysosome over? No. We don't send organelles. We send little chemicals, okay? Little ligands, little footballs, little, yeah, we're sending small things. So, oh, look, the word small. D looks pretty good. Water soluble, which means that it can dissolve in the cell, outside of the cell, and then in another cell. I really like it. I could look at this other one, like branched polysaccharides. I mean, poly means it's big. Branched means it's also big. That, gosh, that's not really the way that we communicate necessarily. We're usually looking for small signals, like you don't have to come in to someone's house with a battering ram or like drive your car through the front door. You just knock. Okay. You just knock, or, like you leave someone a note. You don't, run in screaming at them. That's how cells work too. Let's make this efficient. Let's just do something small. So I really like D. See if okay, great. There it is. And again, B is such a joke. Chromosomes, I'm not going to send you my DNA. Like, this is my cookbook. I'm keeping it for myself. All right. 22 is, the SARI gene. SRY. Okay, Y chromosome, the SARI gene, male fetuses to develop testes. Okay, so that makes sense. And let's look. I'm seeing the answers here. Produces. And what does it produce? So for some reason, I feel like starting at the bottom. The gene is found in the tissues of developing gonads. Gosh, maybe. Yeah, that's no, we know that's not true. Okay. The gene is found only in the tissues? No. What they're testing you here on number D is, on letter D, do you know that every single cell in your entire body, eukaryotic, like your human cells, not the microbiome, every single cell in your body has the exact same DNA. Your testes know how to be eyeballs. Your eyeballs know how to be your nose. Your skin cells know how to be your muscle cells. Your muscle cells know how to be your brain cells. They all have all the instructions. It's just a matter of which genes are expressed. So D's out. The SARI gene produces an RNA segment that is exported from the specific cells and targets the developing gonads. Exported from the cells. That's unlikely, like this is the basics of DNA, or the basics of biology. You got your DNA, and then it's converted to RNA, and then the ribosome translates that into a protein. That all happens inside a cell. We're not exporting from cells to a different cell. It's all right there. It goes from nucleus to ribosome inside of one cell. I hate C. And then it produces a protein that deletes portions of the X chromosome in male so that the male characteristic. What? That's so extreme. We don't need to do that. We don't need to delete. We could maybe block, like you could put an inhibitor on that, but like that doesn't seem, that's not how it works. The Y chromosome can be expressed. It has nothing to do with blocking the X chromosome. That's just not, then you could kind of look up there and you could just be like, yeah, we're not, there's doesn't really seem right. So process of elimination, I like A. Okay. So boom. And interestingly, on this one, the answer was A, and I started from the bottom. So whatever. If you like to start from the bottom up, you like to start from the top down, whatever feels right. The reason why I started with the bottom one here is because it was the shortest. Okay. And oftentimes, I know this is like, I don't really want to say this because it's not always true, but oftentimes the longer answers are more correct and the shorter answers are wrong. Okay. I hesitate to say that. We could look back at all these other ones, and I could try and like look at all of them and just be like, yeah, the shortest answers are typically not the right one. And the longer ones are typically the ones that you want to see. So that's why maybe instinctively I just went for that short one, because I just wanted to rule it out quickly. But maybe the skill is like, go for the long one because you think it's right. But okay, so it produces a protein that binds to a specific region of DNA in certain tissues, which affects the development of these tissues. Boom, perfect. Okay, that's our lac operon, promoter operator. Something's landing on the DNA, and transcription translation make a protein. Okay, that is the deal. Y chromosome male, testes, let's go. All right. As I'm reaching the end here, I'm getting excited maybe I'll have a little bit of extra time to go back and check my work. That's how you want to feel as you're getting to the end of the questions here. Okay. Make sure each question at least gets some of your attention. And then if you have time, go back. All right. Boom. 5%, 35, 50, 95. Okay. The average amount of time spent by a particular in each phase. Oh, this is such an easy one. Not a softball. The time required to complete a full cycle to the cells in interphase. This one is just, you got to know that interphase is G1S, G2. M is not interphase. Interphase meaning like in between, because mitosis is where like action happens. You got like prophase, metaphase, anaphase, telophase, all the action happens. G1S, G2, if you were to look at that cell under a microscope, nothing's happening. Pretty freaking boring. So it's called like the in-between interphase because it's in between the action. And yeah, that's 95 freaking percent. That's most of the time. Bing, bang, boom. Our final one. So exciting. Let's go. Not exhausted at the end, just stoked that you made it. Okay, we got some numbers, 68, 27, whatever. All right, so the amount of time in minutes. Yeah, so when you get these like mitosis questions, you got to rip through them because those questions, like whenever you find an easy question that's your wheelhouse that you're really comfortable with, rip through them so you can spend more time on the harder ones like G protein coupled receptors and cell communication and stuff. Yeast cells grown in a nutrient poor environment. What percent of the cell cycle is approximately what percent of nutrient rich? Okay, so we got nutrient poor versus nutrient rich. We're going to have to do some mental math here. What percent? Oh, Lord. They're giving us, oof, gosh, 168, 20. Gosh, this is going to be rough, I think. The duration of yeast cell in nutrient rich and nutrient poor cell cycle. The cell cycle. Oh, so it's got to be this entire thing. G1S, G2M, and cytokinesis. So are we supposed to like wrap that all up into one thing here? And is approximately what percent of the nutrient rich? Whew. So the nutrient poor, I'm just going to add them all up. And I'm going to be like, oh, that's 60, that's 60, that's 30, that's 10. So 60, 60, 30, 10, 60, 61, 20, 160. So I'm writing that down really quick. Quick mental math. It's approximately 160. Because I don't think we need to be, oh gosh, these numbers are tough. And then the nutrient rich environment, we might set up a ratio. Anytime you want percentages and stuff and like quick math that they'll ask you to do on this, it's not going to be some complicated calculus. It's almost always a ratio. That's why I was helping a former student on a nursing test, and they were asking these tough questions about physics, but it was just a ratio, simple ratio. So X over, 100 equals X over 100, because that's a percent, equals this thing. They're saying the nutrient poor to nutrient rich, right? What percent of the nutrient poor? So nutrient poor, I'm writing NP. Nutrient rich, I'm writing NR. This might be kind of tough to see on my scratch paper here. When we set up a ratio, there it is, X over 100, then nutrient poor over nutrient rich, NP over NR. Okay. And so I'm adding up the nutrient rich and I'm going 23, 48, 25. And then so we've got 25 and 25 and 48 is going to be approximately 50 plus 50 is going to be about 100. Okay. So I'm going to see if this approximation works. And if I have time, I'll go back and do the actual math. It wouldn't be that hard to actually just add it up, but I'm going to see if I can get away with this. So the 160 over 100 means that it's about 160. Okay. So now I know it's either A or D. And since those numbers are so close, I got to double check. If I'm going fast, I'm putting D. That was my mental math, but now I'm going to actually add them up. So 25 and 48, 23 is 71, and 25 is going to be 96. So 96 is our nutrient rich. That is going to go on the bottom of the ratio. On the top of the ratio is nutrient poor, because they asked us the ratio from nutrient poor to nutrient rich. So I'm going to do top bottom and then set it up X over 100, because they're saying X is our nutrient poor, is what percent of our nutrient rich. So we're calling nutrient rich 100 and we're calling nutrient poor the X. And so you line them up just like that. Let's add up our other ones here. 3357 is going to be 90. And then we have 90 plus 10 is 100, so 161. So we got 161 over 96. Damn, that is they are really pushing us to do that. You're gonna have to have a, gosh, I don't even know if they'll probably won't let you have a calculator and stuff. You just got to take a guess. I do think that's closer to 160 than it is to 168. But boom, yes, it's 168. Okay. Yeah, so this yeah, this is a multi-stage kind of a thing. We got to know bio and we got to know math on this one. And then you frankly, like, you have to have a calculator. That's kind of tough. But again, don't worry about the questions you can't get. Okay. Like, if you're not a math whiz, don't worry about it. If you are, give it a shot. If you have time, give it a shot. But yeah, this is like, it requires your knowledge of the fact that the cell cycle is all of these things: SG2, mitosis, G1, and cytokinesis. As long as you know that that's the entire thing, then you know to add them up and then create that ratio. So give it a shot. Okay, thanks for joining.